Swayam Prabha. Digital India. Educated India.
Good morning. Welcome you back to the lecture in continuation with what we have been doing in the previous class. We have been looking at the examples for the TEM, particularly the nanocomposites and etc. And we have seen immediately before closing the previous lecture, we have looked at a case where we can identify even the coordination type, nature, coordination geometry that would reflect on the electron energy loss spectroscopy, where in the near edge of that particular peak you can get information on the chemical as well as the bonding type and the coordination nature, all of these. Let's look at other kinds of methods which you can attach with the TEM and draw your attention to this, where you can even map, since you're able to, through the HRTM, you're able to focus with a particular one single particle. So then you can also try to identify the different elements distributions also. Okay, and that is what is shown here, the elemental and chemical mapping using energy filtering technique. So we don't need to worry about exactly what it means, the energy of the electrons that are coming out. So those are the ones which are being used for this. So it's called energy filter TEM. So it's the same with the TEM instrument, you can make a slight modification. Now, so please have you focus on this particular slide. In the top panel, this is for the Si3N4 polycrystalline material, and this is a 100 nanometer size. You can see the kinds of crystallites, reasonable size within that, about 120 nanometers, and then maybe 50, 60 nanometers in this direction, etc. You can see. Now, if you try to, and this has some impurity or additive as magnesium oxide and yttrium oxide as well. So, if you try to put the elemental mapping, the nitrogen, how well distributed throughout of this, the oxygens are distributed somewhat much different than there is the nitrogen distribution. And then you have the silica distribution, that is the major component. So you can see the distributions of the silica, nitrogen and oxygen, and you would see that this oxygen is coming from these additives. So you have certain level where the oxygen impurities have got doped, etc. You can also do additionally, which is not shown here, for magnesium also you can do mapping. You can also do for yttrium mapping as well by choosing appropriate energy filter electrons into that. So what it gives, you can get a chemical mapping of the crystallites, particles or particle clusters, whatever you have, whatever you focus, and you can see the distribution of the chemical elements in that. Very important. It can be done very well. Okay, draw your attention to the bottom panel. The bottom panel is cobalt chromium alloy, and these kind of alloys again are magnetic in nature, so used in the magnetic memory writing, erasing, these kind of functions. You require a very nice sharp change in the magnetic property so that writing can happen and erasing can happen, and these are used with some kind of a compositional inhomogeneity with some other elements. For example, the COCr alloy is further added with the tantalum, platinum, these kind of things, based on the kind of element that you have, as well as their compositional inhomogeneity, structural inhomogeneity. They all reflect on the properties that you're looking for. The properties you're looking in this is a magnetic property. Now here you can see that the CrCO alloy, the 50 nanometers kind of thing. So size, this is the dimension given, and you have the particles in this core chromium part of that. You can see the mapping for the chromium part and the cobalt part of the mapping of this. And you can look at the ratio of the cobalt to chromium. That is very important. The chromium to cobalt, okay, it doesn't matter. So chromium to cobalt atomic ratios are very important for these Li properties, because their arrangement would provide certain voids in which the other elements can be incorporated, etc., or they provide certain coordination cores where it can be provided, etc. So therefore, the structural property and their ratio of the chromium to cobalt is very important in these aspects. Okay, so this is used, it is found that magnetic properties directly depend on the compositional inhomogeneity of the cobalt chromium films. So therefore, obviously their distribution, the ratio of the chromium to cobalt, all these plays an important role, for which you require a mapping, and that's what you get the mapping in this. Let's look at other kinds of applications. So far we have seen nanoparticles and nanocomposites, etc. What about the biological matters, materials, like for example proteins? If you look at, here 2 examples are shown, but there are a large number is known. One is called the apoferritin, and this particular thing gave the structure at a 2.5 angstrom resolution, which means the residues can be identified, but individual atoms cannot be identified. To get the individual atoms to identify, you need to get down close to 1, 1.1, 1.2. Even closer or lower than 1 is better, so that you can, each atom in a bond can be. But otherwise, if it is above 1.5, the 2 atoms in a bond cannot be separated out. So that's what it is. So here 2.5. 2.5 is roughly, you can separate out this the amino acid side chain, but not individual atoms of that. But anyhow, that is the power that we can do it. And here shown for 2 cases, one is apoferritin, other is a beta galactosidase. Okay, so as I said, the TEM you can vary variety of parameters. So one of the parameters is even the temperature and the cryo system at the cryo level. And you know that we have been talking about, you require a very ultra thin layer of this. I've also talked to you in one of the previous classes about the 3D structures, 3D images of the objects, getting it by varying the angle with respect to the beam, etc., and then meshing, and a large number of, by 1 degree, 1 degree, 1 degree, and combine all of those pictures and make a 3-dimensional object. Second technique I also explained to you at that time was, if you can spread your compound, which is dispersed in a solution, to a very ultra thin level layer, then whatever you have on the surface is each spot will be one molecule, and that is what. So roughly a few nanometers is the protein, and that's what would happen. So how would you do that kind of thing? Generally what you do is a microliter and micromolar. That's still very high concentration, wherein too many molecules will get aggregated when it is evaporated, and then you will get a cluster. Then you will not get a chance to see individual molecules at all. So therefore, in this paper, the speciality is that, though our speciality is to look at the structure, but theirs is a technique development which is called sub-nanoliter. Sub-nanoliter. So how do you get a sub-nanoliter? Microliters you get from the micropipette kind of thing, micropipette, micro syringes. Now, what they used was, you take a microliter at a very low concentration containing maybe 10 power minus 9 nanomolar or much lower, that you can always make, and from that you take a pin and dip the tip of the pin, not the kinds of the size of the pins that we are talking about, much smaller, where the tip cannot be seen by our eyes, and you try to drag on the surface that tip which is dipped into the solution, and that will have a nanoliter level amount of material solution, and that solution is spread by dragging. You can drag in X, Y, etc. Or you don't require Z, you should not, because it will become bigger. So, and then X and Y direction, and that you leave it. And that's what will have ultra thin, and that will have every spot wherever you have after the solvent dries, you will find each protein molecule getting. Now, On that layer, you can find several thousands of molecules within the space of a micron range, within the space of a millimeter range of the space that you have utilized, and that is what is focused. And therefore, now your TEM beam is focused on one particle, a second particle, a third particle, a fourth particle, like that. So you do that. So you take a image for each, and in this case, this is a 2.5 angstrom resolution. It's called 3D reconstruction, three-dimensional reconstruction. In this case, you will get really surprised, 47,209 particles. So in that area, so each one you can focus. Each one you have to focus with the beam. Focus with the beam. The beam should be as sharp. So these are all more of an instrument rather than. So instrumental developments gives a much more higher result structural aspects. So more of a engineering inputs are involved, and then comes is the chemical and structural aspects of coming into that picture. So 47,000 particles, okay, and the other case here, so you can see this particular shape of this, of course, and that exactly. Of course, there are structures, some of the structures known by other methods, and then you try to fit by computational means and see the agreement between the structure that you have and the spot that you got, the picture that you got from the three-dimensional image that you have for each. So each molecule, each spot will be one molecule. So each droplet is one molecule, or each particle is one molecule here. And to get that, you need this technique to be spreading, ultra thin level of that. Second example, beta galactosidase. If you see that, in this case, it is 15,252 points have been focused, and then, so you see that structure like this, and that is exactly this kind of structure. That means what you are seeing here is each particle is one protein molecule. Okay, if your concentration is high, 2 proteins may have been, 3 proteins may have been aggregated in together, and several. So then you will not get the proper information. That is why the technique development. So this particular paper, they call it as a vitrojet, vitrification. So so you ultra thin it and then freeze it. So instead of drying that way, freeze drying. It's called freeze drying, not the normal drying. So you make a very thin coating of that solution with a micro-tipped thing, and you don't do it with hand. All these are done by the electromechanical kind of a connectivity, or where sometimes we use some laser kind of things to move, etc., and then get the spreading of this and then study. Hope that is clear. The example is clear to you how you can get a complicated structure of a protein using the TEM with the fitting. Not only for the proteins, you can do even for the virus. Viral particles are much bigger than the proteins. So the proteins are a couple of nanometers, and the virus particles are several, tens of nanometers, etc. That. Then in those nanoparticles, you have again information that RNA kind of information, protein kind of information, some cell membrane information, some kind of a the polysaccharide kind of information. All these, can we get those kind of things? Yes, depending upon the resolution that you focus using the TEM. So here you have the ultra structure of coronavirus particle in autopsy samples. Formalin fixed and paraffin embedded. So it's put into the formalin and wrapped with a paraffin. Okay, embedded autopsy, olfactory mucosa, re-embedded for the electron microscopy, A, B, and C. These 3 are for these ones. And conventionally embedded autopsy, lung of the COVID-19 patients, this one D. So these you can see the way that these are and the levels at which you can. So if you look at these things where you have the white arrows, the membrane compartments with multiple or single enclosed CV particle, they are white arrows. So you can see here, white arrow here. So these, the white arrow here, the white arrow here. So these are all the ones where the membrane compartments with single or multiple enclosed CV particles. Okay, so that viral particles. So white arrow. Then ribonucleoprotein, so that is RNA part of it. So that these are electron dense and they are partly granule, and these are black arrows. So you can see that the black arrows here. So this black arrows here, this black arrows here. So that inside is the RNA. This inside is the RNA that you have. And if you further look at closer, this is 100 nanometers. If you look at with 10 nanometer thing, you will see that granular structure also for this, as you can see. And surface projection spikes. As you know, the coronavirus has got a lot of spikes, and these are the ones which will recognize the cell and then get attached to the cells, etc. These are white arrowheads. So these white arrowheads is this one. So these are outer. So these are spikes. So these are outer. This is spikes you have. So outer spikes. So you can see each component of the viral components. So if you do it 100 or higher, 500, etc., you will see only, yes, there is a virus particle, but you will not get further information. Here you could get the further information, the particles which are present, the RNA part which is present, and the outer connectivity surface projections, and the biomembrane black arrowheads. Okay, so all these kinds of informations can be obtained for a coronavirus. In the previous example with the protein, you could get the whole structure fitted. Now, by using large number of shapes, size, density, and interior components are there, you can identify what kind of virus is that, because a lot of data is already there available, and then you can utilize, compare. So a little more expanded version is shown here in these things. So these are the ones which are the entanglements, surface projections you call, and you have your the inside RNAs, the other part of it, and the membrane. All these can be seen. So more resolutions have been coming up in the recent days. As I said, can be used for the differentiating what kind of virus, etc., etc. This is the first diagnosed case of the camel pox. This is from the camel virus identified in Israel and studied, etc. So these are the electron microscopy TEM of the viral particles extracted from lesions taken from 4 individual camels and negatively stained with 1% phosphotungstic acid, because you have to get the dark and the lighter gray areas. That's why you treat with that. That examples also I explained earlier when we were talking about this staining part of it, dark field and the bright field, etc. So negatively stained with 1% phosphotungstic acid. So those are more of instrumental details. You don't need to worry. Note that the morphology and the dimensions of the virus particles correspond to the morphology and dimensions of the orthopox virus. So these ones, if you see, these are all belong to that orthopox virus whose details were known. Okay, the F is this one, and this one, for comparison, a TM of an ORF virus, the alternative, the negative staining. After that, you can see that dark gray, but with difference in the shape, and different is the interior as compared to this. So these will have one type, this will have another type. So that means you can differentiate the viral particles from one type of virus to another type of virus as well. So there is a huge morphology difference between the ORF virus, which is shown here, and the PPV virus, which is shown in these family of that. So using the TEM, you can definitely identify the types of viruses. An example I showed you in the previous slide, even the coronavirus, where reasonable levels have been identified, the exterior attachments, interior RNA, and things like that. Further pictures are coming up even in nowadays with a greater resolution. So if you go into the literature, you will find even better than that as well. Okay, so we have looked at a large number of examples of variety of things, nanoparticle and nanocomposites. They're identifying different chemical regions, identifying different the coordination kind, all those things you can map. So, and then it can be extended, these techniques, to the protein structure determination, viral particle detections, etc. So all of these. Okay, so quite well depth that we have treated in terms of applications. The course is mainly for applications, so therefore we go into all of those. Okay, so let us look at the last technique under this microscopy that I would like to cover. Is not that really the last technique, but it is for this course, for this particular topic. Yes. And so this is the scanning electron microscopy. I have already introduced a couple of classes before, where you have the electron backscatter and secondary electrons. These are the ones which you use. In fact, they use both of these in many of the techniques. And the detectors are set in such a way to capture the backscattered or capture the secondary, all these kinds of things. Show you in a while. So this SEM type electron microscopy is focused high energy electrons, and as I said, you can get the sample morphology, you can get chemical composition, you can get chemical structure, all these kinds of things you can obtain. Okay, so you can use the all kinds of techniques. So that backscattered is dependent on the mean atomic number. So the greater the backscattering, then the main thing will be smaller or weaker or grayer, and the other one is darker, and vice versa. If you filter off the backscatter, then you will become bright, the darker. So you can try to create those from there. These are backscattered is a relatively high energy, so that means you require a very high resolution in energy measurements to get the picture, to get the image well. Okay, and whereas secondary electrons are not as the higher as that of the backscattered one. So somewhat. So therefore your detectors also change. Not easy to have energy dispersive techniques, brighter, etc. All this. So this can give you a thing from the millimeters down to the nanometers. Okay. So with a very good resolution in the lateral, means XY kind of a thing, to some extent Z also you can get, but you don't see the transmitted, but that you will see in the TEM, but you will see some level of depth also, as if it looks like a three-dimensional, though it is not completely, because you don't see full transmission. You see some depth entered in, and then give the secondary electrons. That's where. So you can get scanning mode with a high lateral resolution, broad magnifications, and large depth field. Okay. So all these, if you combine them, it looks as if it is the total object kind of thing. So depending upon the nature of the sample analyzed, SEM can analyze a sample non-destructively, allowing the analysis of a single material by multiple techniques. So these things, high resolution images, it can give chemical composition also, you can identify the extent and the distribution, both the quantitative and the distribution parameter. Phases, what kind of a phases you have in a multi-phase materials, because some crystalline materials can be in more than one phase. So therefore you can identify what composition of that phase versus what composition of the other phase. Secondary electron backscattered electrons, etc. So just to show that an example is shown over here, as if you're seeing through, but you're not seeing through from TM, you're seeing still a top with some level of depth that you can see here. And this is exemplified depth field yielding a characteristic three-dimensional appearance. You can see as if it is a three-dimensional kind of thing. So useful for understanding the surface structure of a sample. So this is exemplified by the micrograph of pollen. So this is the picture for a pollen, and you can see different kinds of particles you have. These the spherical balls type with these needles projecting, and these shell types and other things, the smaller spherical ones, and some triangular, huge number of things in the pollen. That's where. So a wide range of magnification is possible, and about 10 times and to about 250 times magnification as compared to the light microscope. So you can get all of these particles. If not the interior structure or that morphology, you can get this. Okay. So these are all done by the scanning that you can easily read, and the specimens are taken at a very kept at a high vacuum. Now there are the SEM instruments developed with somewhat much lower vacuum. So with the lower vacuum, these are called environmental SEMs. So you can use somewhat lower vacuum with some kind of a pressure conditions, other kinds of environmental conditions you can maintain, and then look at or you can maintain the temperature conditions like cryogenic. Just like cryo-TEM, you have cryo-SEM and environmental SEM. So of course, where the resolution is somewhat less, but you get a broad range picture of the images that you can get. So like here is a broad range. All kinds of particles have been identified. You can get quantifications of these as well. You may not get interior of each of these in more detail. But in some cases, you can go to the nanometer level, not in all, but in some cases, in special cases, you can go even up to a few nanometer to 1 nanometer level, but somewhat less in number in that. But still, it gives the surface as compared to that of the TEM case. Okay, just now I mentioned to you about you can have a different kind of detectors to measure the secondary electrons, the backscattered electrons, etc., etc. Just look at the principle. We have already looked at the principle for the TEM quite well earlier. Very similar kind of features can be used here, like for example, electron generated system, which is called electron gun. Okay, it's just a working principle only, nothing more than that. And then the apertures, then beam, and then you have a lens which is focusing, then again apertures, then lens, secondary lens focusing. There are reflection coils, etc., which is the magnetic ones, because of you flow the current then through them. And then you have the other lens apertures, and you can get the reflection backscatter for the detectors or secondary electron detectors, and of course the sample is there. So you can get the secondary backscattered ones, secondary electrons, and a lot of other things will also come, like the X radiation, etc., all of these. So you can attach other detectors as well. Their angles will vary. So everything will surround that your sample. In all directions, these different guys are sitting in order to measure various aspects of this. Okay, so having looked at the very basics, I already introduced earlier, again reintroduced now at the time, and the principle, we can look at some examples of these. Okay, so the examples of these, again, I will not go into as many deep examples as I have already explained for TEM case, because they are very similar in interpretations, etc. Though somewhat the features are different and some resolutions are different, but can be used for the similar kind of purposes. For example, for the nanocomposites. Here, this is a nanocomposite. Draw your attention to this. Is a glass fibers reinforced or bamboo fiber reinforced epoxy. So you take epoxy, is a polymer, and to that you add certain bamboo fibers or glass fibers, and then you reinforce, you make a composite, and the composite will have a different kind of a structural features or mechanical feature or flexibility features as compared to the simple epoxy kind of thing. Okay, and this paper has got all both for the glass fiber and bamboo fiber. The example that I've shown is only for one of them, the bamboo fiber with this. And then when you treat these bamboo fibers and use this one, the alkali treatment and without alkali treatment. So this is a without alkali treatment, this is a with alkali treatment. And anyone can say just by looking at it, here these are all in random kind of thing, here is all arranged kind of thing. So that means more crystallinity has been developed as compared to. So you have a less crystalline or more powder characteristics and more crystalline kind of a characteristic. And that brings a change in the flexibility properties, the flexor properties of this. That means the epoxy, which is reinforced with the bamboo fibers, not normal fibers, the fibers which are treated with the alkali, they give more crystalline nature. And this strength of the flexibility properties of this, flexural properties of hybrid composites increase with glass fiber content. It also increases with the bamboo fiber components, but which are treated with the alkali. So these properties found to be higher when alkali treatment of the bamboo fibers were used in the hybrid composites. The elimination of the amorphous hemicellulose with alkali treated, leading to higher crystallinity of the bamboo fibers with alkali treatment, is the one which is responsible. So what is responsible here is basically the fibers which are treated with the alkali. When you treat with the alkali, then they will diminish the non-crystalline matrix of this epoxy part of that, and thereby or hemicellulose, all of these, thereby you increase the crystallinity, and thereby you increase the composite properties of the composite. better, increases their composite parameters of this. So strengths, etc. they are making. So I think we have some more examples coming up, a few more, but not as extensive as we have done in case of the TEM, but we will try to take up in the next class and complete that one, and then we will look at the overall course of what all we have gone through and how we have gone through, etc. These will come up in the next 1 to 2 classes. We can try to wind up the whole methodologies that we have, all the techniques that we were trying, analytical, spectral, and the last part of the microscopy techniques, and we'll cover in that. Thank you very much.